Hi and welcome everyone to this video. Here I'm going to explain as briefly as possible the research I presented to ESMO Targeted Anti-Cancer Therapies 2020. This research, Proteomics in Advanced Melanoma Samples Treated with Anti-PD-1 Immunotherapy, unravel a group of patients resistant to therapy, is framed within an industrial doctorate that is a collaboration between Biomedica Molecular Medicine and the University Hospital of La Paz. This presentation will have a few shortcuts to prevent you from getting lost. First, I'll talk about the background, all the research done to get to this study. At some points, I have to stop to clear out the most important methods. Then I'll present the results we got at this point, and finally I'll talk about the experiments ongoing at the moment and the future steps. So let's begin. This research began a few years ago when we downloaded the data from the Cancer Genome Atlas portal. There was data from 472 skin cutaneous melanoma samples. We downloaded the clinical database and the mRNA-seq dataset. We selected those missing values in the mRNA-seq dataset that had a variance higher than 2.5. And finally, we got 2,971 genes. Then we build a probabilistic graphical model. But wait, I need to stop here and explain a little about probabilistic models. Probabilistic graphical models, or PGMs, are undirected graphical models obtained as a result of minimizing the Bayesian information criterion, BIC. Technicalities aside, PGM's results are this type of graphs, something like a network where each box represents one node, that could be a gene, a protein, or a microRNA, and each line, edge, joins two nodes with the related expression in the dataset. Now that you know a little of PGMs, we can continue. So we build this probabilistic graphical model with the 2,971 genes selected. Then we performed the gene ontology. We saw that the gene expression grouped genes in branches with the same gene ontology. This way, we proved that genes involved in the same function had a related expression. We saw 21 of what we call functional nodes. Once we had the probabilistic graphical model, we studied by searching the informative layers. These are small database within the original dataset with genes selected by sparse k-means and patients clustered by consensus cluster. But wait again. What are these sparse k-means and consensus cluster? Sparse k-means is a type of k-means that simultaneously identify the clusters and the important clustering variables. Consensus clustering outputs show the optimal number of clusters to divide the matrix with all the patients and the genes obtained by k-means. Knowing that, you might understand the idea of informative layer, which is finding all the important information present in a database through deconstructing it in small datasets. Using these methods, we identified 112 genes within the informative layer. These genes divide the dataset in two groups. These two groups had different expression of immune nodes, but there are no difference in overall survival. In this point, we thought that maybe there are no difference in survival because these patients did not receive the appropriate treatment, as it could be an immunotherapy. We talked to the oncologists specialized in melanoma, and they told us that it will be interesting if we could transfer these groups to a dataset of patients treated with immunotherapy. So what we did was go into geo.set and look for some research of melanoma patients related with immunotherapy. We found Hugo et al. Database, which had 28 samples treated with anti-PD-1 immunotherapy. Some clarification about anti-PD-1 immunotherapy must be. The immune system has the ability to attack cancer, but tumors are smarter and had lots of mechanisms to evade the immune system. One of these mechanisms is the PD-1 PD-L1 blockade. When PD-L1 binds PD-1, the cancer puts the brakes on the immune cells, blocking its attacks. The anti-PD-1 treatment operation is unblocking this ligand, reactivating the immune cell attack. So we hypothesized that patients with low expression of immune genes had a lower activity of immune cells. So these patients will not have a good response to treatment, because even the treatment allows the immune system to avoid the PD-1 blockade. If the immune system is not active, it will not attack the tumor, obviously. So back to the validation dataset, we transferred the informative layer classification and checked the overall survival of these groups. Here we found that there is a group of patients that are responders to anti-PD-1 treatment. This finding will possibly allow knowing which patients are going to respond to the treatment. This is important enough to continue with the hypothesis. So that's what we did in this research. We want to validate our discoveries in our own cohort. With help of Grupo Español Multidisciplinar del Melanoma, 53 patients treated with anti-PD-1 immunotherapy were recruited. Samples were collected from 2016 to 2019, and patient characteristics are described in this table. First of all, it is convenient to say that 23 patients were treated with pembrolizumab and 23 patients were treated with nivolumab, two different anti-PD-1 immunotherapies. 68% of the patients were males. 88% of the samples were cutaneous melanoma. 51 were primary melanoma, 19 were nodes, and 30% of the samples were metastases. It is remarkable that 9 patients had complete response after the treatment. And finally, there were also data from LDH, metastasis locations, and ECOG. With these patient characteristics taken into account, formal fixed paraffin-embedded melanoma samples were analyzed using high-throughput proteomics approach based on mass spectrometry. Data regarding protein expression in melanoma sample are scarce, so we took the opportunity of having 53 samples to analyze them by high-throughput proteomics. Proteomics data was analyzed using probabilistic graphical model, sparse k-means, and consensus cluster algorithm. The resulting network had 8 functional nodes, and we identified an informative layer of 107 proteins that divide the dataset into groups, one of them with better relapse-free survival. We also start the process of RNA-seq of 2000 preselected genes through CCAP RNA choice proofs. These experiments are currently running at the Parque Científico de Madrid. We expect to find the relation between this informative layer from the TCGA and the response to treatment. To look for that, we will use probabilistic graphical models, sparse k-means, and consensus cluster. We hope to present all this data at ESMO 2020 in Madrid. So we reached to the end of the video, but I want you to take home some messages. Mechanisms explaining differences in anti-PD-1 treatment efficacy have not been elucidated yet, but our group had found an immune signature that predicts response to anti-PD-1 immunotherapy. Proteomic analysis showed different expression patterns in patients treated with anti-PD-1 immunotherapy, and RNA-seq analysis will shed light into the efficacy of immune signature previously described. Thank you for watching this entire video, and we are Molecular Oncology and Pathology Lab and Biomedical Molecular Medicine. We have collaborated with all these institutions in this study, and here is my mail in case you have any doubt. Thank you again, and see you in future videos.